Aerotolerance, determining the oxygenic requirements for growth in bacteria. You begin the aerotolerance test by gathering 4 test tubes from a hot water bath. This test must be inoculated with the agar in molten form. If you are unable to use a beaker of hot water to keep the agar in molten form, you may want to just get 1 tube at a time, and after inoculation go back and get the next tube. The agar used for the aerotolerance test is BHI agar, which stands for Brain Heart Infusion Agar. This is an enriched media that promotes the growth of anaerobic bacteria. Inoculation of the BHI can be a little bit tricky. You are inoculating the agar with a liquid culture. However, you do not want to introduce air bubbles or oxygen into the media, since we are trying to grow organisms in anaerobic conditions. Use a sterile transfer dropper to draw the media up. The droppers being used in this laboratory are graduated, so you know exactly how much of the culture is being drawn into the dropper. Carefully release some of the liquid culture into the top of the tube. Then slowly push the dropper down into the medium and continue pushing the culture down into the molten agar. Just before you reach air bubbles in the bottom of your dropper, you will want to remove the dropper from the media and finish the inoculation on the surface. This is done to ensure that bacteria will be dispersed throughout all of the molten agar and will not remain at the surface. Here, a plastic dropper contains the culture that's going to be used to inoculate a molten agar BHI. Notice the tip of the dropper rests towards the top. As the fluid comes out, the dropper is immersed into the media. Just before air bubbles are released, the dropper is pulled out of the agar. If air bubbles are present on the surface of the agar, they can be easily removed with the dropper. Shown again is the same process. After inoculation, the bacteria needs to be mixed with the molten agar. This is done by carefully rolling the tube between your hands for about 30 seconds. This will ensure even distribution of the bacteria through the molten agar. Immediately after inoculation, all of the tubes of molten agar that have been inoculated should be placed in an ice bath. This will cause the agar to solidify quickly, suspending the bacteria throughout. Interpretation of results. Agar deeps are used for inoculation to create an oxygen gradient from the top of the tube containing the highest concentration of oxygen to the bottom of the tube containing no oxygen at all. Since bacteria were suspended throughout the entire agar deep, they will grow where oxygenic requirements are met. This can be seen or observed after incubation by cloudiness appearing in the media. It is easiest to interpret your results by comparing them to an uninoculated control. Now shown is E. coli. E. coli is a facultative organism. Facultative organisms are capable of both aerobic and anaerobic respiration. E. coli is also capable of fermentation in anaerobic conditions. Facultative organisms are capable of switching their metabolism from aerobic to anaerobic or vice versa, depending on the oxygen that is present in the environment. Here, E. coli in anaerobic conditions used fermentation with gas production. Gas production is obvious by the presence of the fissures and cracks in the media. So much gas was produced by fermentation in this particular tube that the media actually slid up the tube and into the cap. Upon lifting the cap, you would see that E. coli was present on the surface of the media. This indicates that the organism was capable of growth both in aerobic conditions at the top of the tube and in anaerobic conditions all the way to the bottom of the tube. Now shown is Clostridium. Clostridium is an aerotolerant organism. Aerotolerant organisms are only capable of anaerobic metabolism. However, they are capable of growing in the presence of oxygen. They simply do not use it in their metabolic processes. The arrow is pointing to the end of the line of growth towards the top of the tube in Clostridium. Clostridium will grow in the bottom 3/4 of the tube, but will not grow near the surface. This is because although aerotolerant organisms do not use oxygen in metabolism, high concentrations of it can be toxic to the organism. Now shown is Pseudomonas aeruginosa. Pseudomonas is an obligate aerobe. Obligate organisms are required to grow either with oxygen, as in an obligate aerobe, or without oxygen, as an obligate anaerobe. Pseudomonas should only be observed growing on the surface of the agar. Finally shown is Bacillus megatherium growing in the agar deep. Bacillus most of the time will grow as a facultative organism. However, in certain conditions it will grow as a microaerophile. Microaerophiles are organisms that require oxygen for growth, but the oxygen must be in very low concentrations. Too much oxygen will be toxic to the organism. However, it cannot grow in fully anaerobic conditions. Microaerophilic organisms are difficult to culture and often require specialized equipment that will alter CO2 and oxygen concentrations in the environment. The aerotolerance test is important for understanding the oxygenic requirements of different bacteria. This allows an understanding of the different growth requirements and ecological niches that some bacteria occupy.